response post microscopy determinate local piezo property. For organic materials, determinate this property is useful too, and it's possible to detect because organics molecular and organized microstructure have low symmetry. For piezoelectric response need have structure with non-center symmetry. General two modes is contact and tapered mode, and possible to detect electrostatic interaction, additional electrostatic interaction in this situation. Is possible to determine what is charge distribution, what is phase potential. Magnetic property possible to detect if use magnetic cantilever. Piezo response if we apply additional voltage current, and friction post microscopy possible to use for investigate mechanical property, friction coefficient, for example. Okay, piezo response. How we organize piezo response post microscopy? Parallel determinate topography. We apply additional voltage on cantilever, and for piezoelectric material, we have additional deformation. This is deformation, same topography effect. For separate topography and piezo response, we use not a DC, we use AC voltage, and this piezo response absolutely correlate with frequency what is we apply, and simple electrical scheme is possible separate topography and real piezo response. Here you see example topography and piezo response. This is image obtained on the same time, but you see absolutely different. On topography we have scratch, quality of sample surface, real sample surface, real morphology. And for piezo, we see additional domain, what is not appear absolutely on topography, and this is domains corresponding orientation of polarization. If you have phase transition or any other process, this domain structure can change. And during phase transition, we have change dielectric permittivity, and this is effect is electrostatic, and this is effect is possible to detect too. This is a macroscopic dielectric permittivity, and this is macroscopic. It's relatively little shift because this is surface, it's not bulk process. Okay, we have always electrostatic effect because we have dependence capacitance electric. Piezo response, piezoelectric coefficient, it's a complex effect, and characterize several piezoelectric coefficients. And this effect is dependent what is orientation of our sample and the electric field relatively. Most simple situation when polarization perpendicular to apply electric field. In this situation, if you have electric field parallel polarization, in this situation we increase size of domain, and if opposite is decreased. You see it's a topography effect. But as I'm saying, when we apply AC voltage, in this case, it's possible to separate topography and the piezo response. But we have two different type of deformation of cantilever. This is vertical deformation, and we have one component of piezo response, and torsion rotated cantilever around the main axis corresponded another component of piezo electric coefficient. And this is two channel in our system of in photodiode, and we can detect on the same times these two components. Normally, we have on image not only topography, this is fibers, topography, we can see morphology, diameter, and so, and piezo response. We have area with signal in plane and out of plane. It's relatively correlated, but it is different orientation of polarization. And if it's not enough, because for piezoelectric coefficient, for determining piezoelectric coefficient, need several image. But it's possible if you rotate a sample, is possible to detect free component, and in this situation, it's possible to estimate what is piezoelectric coefficient in your materials. Distribution of particles, because our method is very local, and resolution, of course, is not 2 nanometer, as I'm saying. 2 nanometer is maximum for topography. Piezoelectric measurement is have a relatively less resolution, but 10, 20 nanometers is possible. And if particle, piezoelectric particle or not piezoelectric matrix distributed, we can see distribution of particle corresponding what is size or what is microstructure of these fibers. This is another example of fibers. For organic materials and biology material is very often not present piezoelectric property at the beginning, because microdomain or layer structure organized opposite orientation of polarization, and in this case we detect at the beginning piezo response is 0. But if we apply voltage more 30, maybe 100 volts, it's possible to switch and or order it domains and appear more and more strong piezo response. This is very, very well-known effect for ferroelectric. If you have one orientation of polarization, if you apply voltage, you can switch and obtain hysteresis loop. In IFM, atomic force microscopy and piezo force microscopy, is possible to do local hysteresis loop. This is a scheme. We use this because we have additional electrostatic effect, and in this situation is better to measure without electric field. But macroscopic, as I remember, we use this scheme. And this is another example. This is PVDF. It's very famous piezoelectric, ferroelectric organic polymers with strong piezo response. and the domains what we create depend from time, voltage, obtained hysteresis loop, dependence, I think, voltage and pulse. Okay, nano tubes. Another interesting organic materials with microstructure. And 20 years ago, we started first time grow dipeptide nanotubes. Now in dipeptides, maybe 10, 20 different dipeptides possible to grow like tubes. But interesting situation with dipeptide tubes show very strong piezo response. You can see here same image and atomic force microscopy, and this is piezo force microscopy. Okay, what is correlation between same image and IFM? Practically, atomic force microscopy very close to maximum resolution of SEM. But in atomic force microscopy, we not only reconstruct real morphology and real topography of our material, but we have visualization of physical property. In this situation, we have piezo response visualization. Okay, again, piezoelectric properties depend from orientation, and for dipeptide nanotubes, polarization oriented along tube, and here you see very clear if tubes are oriented like this, we have strong in-plane signal, but no out of plane. It is very logical because in this geometry we don't have piezo response perpendicular tubes. Piezo response depends from angle. Here is angle dependence. If polarization in these tubes oriented perpendicular along tubes, for obtaining D-free coefficient, need to organize the situation perpendicular, need to measure along tubes, but in this case need to grow with these tubes. It's possible, and you see here strong piezo response when tubes oriented perpendicular to face. Okay, and our estimation what is piezoelectric coefficient, it is very relatively strong. Of course, this is less than a famous PZT, for example, field, but it is relatively strong. For organic materials, similar to PVDF. Okay, what is main problem for investigation by atomic force microscopy organic for all method? It's not so stable with temperature, and it's relatively soft material. We have during atomic force microscopy measurement mechanical interaction, and recommendation use more soft cantilever. Temperature dependence is possible to measure in atomic force microscopy, but technically for IFM temperature 200 Celsius is high temperature, because cantilever, silicon cantilever sends to temperature some destroy, decrease resolution, change conductivity, and this is not so. It's not possible to do temperature measurement with high temperature. But for organic, very often phase transition less than 200 Celsius in dipeptide tubes, we have phase transition about 150 Celsius. This is structure phase transition, it's new structure, and you see when we hit piezo response is absolutely disappear because it's transformed in another phase. But this is phase, okay, this is measurement is correlated with macroscopic measurement, but not zero piezo response. When we apply voltage on the high temperature phase, we see piezo ferroelectric property, and it's possible to create new domains. As you see here, blue is initial. We don't have switching because initial phase is only piezo electric, but after hitting, we have very good ferroelectric hysteresis loop. Okay, this is conclusion about for dipeptide. We have very strong piezo response. Amino acids. If for dipeptide we have two amino acids constructed in one molecule and organized microstructure. But for simplest organic molecular is very important amino acids, we can okay, it's different amino acids is natural important for our life. We have more amino acids is natural, but it's only 20. And organized is protein is important for organized membrane, some process in cells, and so on. So in this case, amino acids is important. Organized proteins as different ordering in amino acids. But we interested in simplest amino acids, and as I'm saying, piezo response appear when crystal structure don't have center symmetry, but practically all amino acids have structure without center symmetric, and it can be show piezo response. It's very easy to grow. This is microcrystals. This is morphology, and here you see very clear appear negative and positive contrast corresponding to different orientation of polarization in amino acids. This is glycine amino acids. In our condition of crystallization, we can see distribution of particles and different contrast on different grains. It's corresponding different orientation of polarization. Here is more systematic. As I'm saying, we have two different components, and here orientation of polarization is perpendicular to sample surface, and we have not practically in plane component. This is more oriented of crystal. Interesting effect we find in this glycine microcrystals switching ferroelectric property. And here is hysteresis loop. It's possible to see change contrast after apply voltage. This is hysteresis loop local. It's not macroscopic. All, as I'm saying, all our measurement is local, and hysteresis loop and piezo response is local. Here is same situation, but what is interesting in organic, in this organic microcrystal, our domain structure is very asymmetry. If you see here, this domain topography, but this is domain structure is this domain oriented along this axis. Problem for atomic force microscopy and piezo force microscopy, we don't know what is orientation, because it's not possible to do some local determine what is orientation. It's not no any method for this. But after polling, after apply voltage, we see appear new domains and domain boundary. How we explain this situation? Our crystal have orientation of polarization along main axis of microcrystal. When we apply voltage, our electric field in the sample have different orientation corresponding to the contact point, and you see very asymmetric. And if when we apply voltage more than capacitive field, polarization oriented is like this, and in this situation we see different orientation of polarization in different domains with opposite polarization. And yes, experimentally we see this situation here at contact point, and new domain grow in this direction. We have very strange domain structure, because it is very asymmetry grow domains. Yes, it is. But of course, this is correlated with orientation, crystal structure, microcrystal structure.
Excuse me, Dr. Igor.
Yes.
Time is getting over. Could you please finish in another
Okay.
5 minutes?
I have little, but I can finish few minutes. Okay.
Yeah. Please. Yeah. Thank you.
Okay.
Sure.
Thank you. I'm sorry.
No
Okay.
problem. That's okay.
Okay. Modeling. Few words about determinate piezo response coefficient. For this is example for another. This is valine. It's another amino acids. But question is, what is piezo response? This is a little problem, because we are measured local, and this is our signal depend what is orientation, yes, and what is statistic. But it's not possible to obtain big statistic, because we are measured locally. If we scan big size, in this case is less, more statistic, but is less resolution. But it's possible to estimate, use standard software, and determine what is piezo electric coefficient in material. But error is relatively big, so 10-20%. Okay, this is example of biology materials. For biology materials is problem, because we don't know what is chemical composition only from literature, but it's possible to detect interesting ordering microstructure in piezo response and ferroelectric property, and now it's neuron is possible to do some visualization piezo response in neuron cells. Friction coefficient. Okay, we have in atomic force microscopy contact and the mechanical interaction, and this effect is possible to use for characterization mechanical property of material. And from this very simple formula, you can see we can determine friction coefficient and adhesion force. And here it's example how possible to do this. And if you apply and destroy, if you apply more force, you can destroy and investigate what is stability of this material. This is another mode. This is like lithography, but it's possible to investigate for mechanical stability and how interact cells for different substrate, and how we apply normal force and scanning and press on the lateral scale and move cells and determine what is adhesion. And the conclusion is for our materials, it's relatively strong adhesion, and with composite with with carbon nanotubes, adhesion a little more. Okay, conclusion. okay, I don't show all result, and okay, this is impossible, but atomic force microscopy is a very useful method for investigate physical parameter, physical property of nanomaterials and organic materials too. Thank you very much for attention.
Thank you very much, Dr. Igor, for a very interesting presentation. Now the session is open for questions.
Hello, can you hear me, sir?
Yes, yes.
Whether unsaturated compounds like which is having a double bond will have any influence with this piezoelectric force microscopy PFM?
What? Sorry, what?
I'm talking. You are discussing about the analysis of organic compounds, right?
Yeah, no, not only composite. I prefer pure, but yes, composite. Yes,
Yeah, if that organic compound is having some double bond, which will have more electron density, what kind of PFM we can expect?
No, because we don't involve chemical process. In this case, it's relatively difficult. I think you ask me from chemical
Yes.
side. What is because I'm not a chemist, I am physicist. In this case, I'm interested in only mechanical and physical property. But what is the situation here? On my opinion, this situation. What is the chemical bond in sulfate? Okay, we in this method we are detect only physical property and chemical property on the surface. On my opinion, in a situation with different chemical bonds, we can detect surface potential, different surface potential. This is not piezo response, but this surface potential Kelvin mode is one of mode, and I don't show this result here because we did this, but it is not so easy to understand because need to very clear understand what is exactly chemical, what is situation on the surface. But it is possible. On my opinion, different chemical bonds reflect on surface potential. Yes, this is effect. And this is not for piezo, because piezo, this is contact. We absolutely not modify surface. Yes, but it is piezo response. This is bulk property. Yes, this is crystal structure. But Kelvin probe, no surface potential mode. It is. I think it is possible. This is possible, and very often we see very good contrast in surface potential, and we don't understand why. No, because it is very, very sensitive for oxygen situation and so on. This is chemical process. Yes, this is.
Thank you, sir.
PFM, I think it is not related to chemical process. This is crystal structure. This is
okay. Thank you very much, Dr. Igor. Any other questions from the audience? Students must be asking some questions. I have one question, Dr. Igor.
Yes.
So because the sensitivity, the spatial sensitivity being so high, like in the nanometer domain, so your sample should be very flat, isn't it? The sample quality, the surface flatness of the sample, should it be very good to do this measurement in PFM?
Yes, this is very good question. Okay, I'm not say about this, but for for any measurement, because contrast is more better to do. If you have contrast, it's more better to do visualization. Yes, but topography, morphology is if it reflect on any property, not only piezo. It's electrostatic, magnetic property. It's effect too. Question is, if you have less roughness, in this case, effect on the characterization any physical property is less. In this case, you're right. It's more better to have a more flattened surface. Yes, you're absolutely right. But this is depend from sample. If you have film, you don't. It's not possible to modify morphology because this is grain structure of your film. Yes, it is not possible. Okay, we measured situation in film without any treatment, but single crystal, yes, it's possible to polish. But as grown very often, surface of single crystal is absolutely atomic flat. It is depend. But if you polish, you may modify surface. In this case, maybe it's not so. This always depend from situation. But in IFM, maximum displacement 5 micron. Yes, IFM like smoothing surface, but 5 micron it is not so smooth. Yes. But yes, if you have, it's not only depend what is step, it's depend what is tilt. Yes, angle. This is important. But it depends from situation and depend from your signal. Yes, always in all IFM and PFM, EPA morphology. Yes, it's not possible to remove absolutely. I hope I'm answered. Yes or not? Oh, okay. What is?
We wish you were here personally, but we understand the constraints. Thank you once again. Thank you.
Thank you for attention.
Thank you, Dr. Igor, for your valuable lecture.
Okay, thank you.
So the next talk is by Professor Madhu Thomas.
Participants,
Madhu Thomas is here. Please check. Yeah.
Participants are hereby informed that the talk titled A Roadmap of Two-Dimensional Materials for Magnetic Storage Applications by Dr. Aravind Puthirathbalan is postponed to the 4th of January. Please note, the talk titled A Roadmap of Two-Dimensional Materials for Magnetic Storage Applications by Dr. Aravind Puthirathbalan is postponed to the 4th of January.
now we have an online session on nanomagnetic materials, a coordination chemistry approach by Professor Madhu Thomas, Associate Professor, Addis Ababa Science Technology University, Ethiopia. I request Professor Reggie Philip to introduce the speaker.
Good afternoon, Professor Madhu. Are you online? Hi, Igor, are you still online? We are still seeing your presentation. Could you please stop presenting, Dr. Igor? Good afternoon, Dr. Madhu Thomas.
Good afternoon.
Yeah, I'm Reggie Philip, chair of the session. So I will briefly introduce you to the audience. Dr. Madhu received his PhD. Yeah, Dr. Madhu received his PhD in inorganic chemistry from Mahatma Gandhi University, Kerala. After postdoctoral research at the Karlsruhe Institute of Technology in Germany, he moved to the University of Guelph in Canada as a research associate. Currently, he is an associate professor of inorganic chemistry at the Addis Ababa Science Technology University in Ethiopia. His research interests concern the design, synthesis, and full characterization of primarily paramagnetic polymetallic complexes of transition metals and lanthanide ions with polydentate ship-based ligands, as these have interesting magnetic properties.
Okay,
Dr.
sure.
Madhu, you can start now.
All right. So let me share my slides.
Sure.
Can you see my slides? Hello?
Not yet.
Not yet. I think the previous speaker is still online. Looks like
Looks
my
like.
screen is reading like that.
Yeah, yeah. Okay. We will see how to solve this problem.
All right.
Dr. Madhu, can you try now? Looks like it's all right now.
All right. Sure. Can you see the slides now?
No.
No.
The previous speaker has removed his slides. He is not presenting anymore. Maybe you want to log out and log in once again?
Sure.
Yeah, you can try that. We'll see.
Let's go to now.
Yes, the slide is coming
Got it?
now.
All right.
Yes,
Okay.
we got
All
it.
right. So I can start now.
Yes.
All right.
Yeah. Thank you.
So good morning, good afternoon, everybody. I am Madhu Thomas. I am from Kerala, basically. I love to speak in the seminar there in Kasaragod. I am long away from Kerala. Now I am in Ethiopia. Today I am working. I have a small research group here with the 4 students, and 3 of them are graduated recently with a PhD, and one still working. And actually, I am working on molecular magnetic materials made up of transition metals and lanthanides. Actually, the research interest is actually I want to make a single molecule with showing magnetic behavior. And actually, I am taking coordination chemistry approach for making this molecular magnetic materials. Primarily, I am using shift-based ligands. It is very easy to synthesize. There are 3 different ways to
Excuse
get
me,
molecular.
Dr.
Pardon?
Could you please go to full screen? If you press F5, you can go to full screen.
Okay, F5.
Yes.
I want to go to F5. All right. Now it's okay.
Yes, yes, it is.
Is it
Yeah.
okay now?
Oh, yes, it is full screen now. Please continue.
All right. All right. Okay. Actually, I am basically a coordination chemist, and I have the coordination chemistry approach to make this molecular magnetic materials. And we have 3 different way of to get molecular magnetic materials. One is by transition metals. Later I started using lanthanides because it has electron-rich F orbitals. And another very easy way I approach these spin crossover compounds of transition metals, especially with the iron 2. The single molecule magnets are they are nanomagnets. They are showing all the properties of the magnets, just like a slow relaxation of magnetization at molecular level. And for a magnet, the hysteresis is also a needed requirement. And we are looking for materials having all these properties. And the potential applications of this SMMs, like all magnetic materials, they used in higher density storage information. And these are some applications of SMMs. They are using in magnetic drug delivery, spintronics, MRI contrasting agents, high density information storage, like I discussed before, and quantum computation, as well as ferrofluids. So actually, we are the people playing with the spins of the electrons. So just like I showed here, here the spins are randomly oriented, and then on applying field, we can have ferromagnetic, antiferromagnetic, ferrimagnetic, and spin frustrated systems. We are trying with all those things. And usually our approach is to prepare ship-based ligands. And it is very easy to condense to make a primary amine and an aldehyde, and make the condensation by the removal of the water. If you see here, this kind of condensations are very easy. And it is easy to tailor the ligands. You can see here R1, R2, and R3. This R1 and R3 can be changed according to our interest. And just like we are in coordination chemistry, we are calling it as varying the ligand fields. So actually, the first row transition metal molecular materials, I gave it to one of my PhD student, Fikri. He graduated recently. And before that, I had some previous experience with the Professor Pavel's group, making this kind of ship-based ligands and making molecular magnetic material. So if you look here, an aldehyde, this is aldehydic part, and this is amine part. When we make the iron compound, we got a iron to a V-like compound with the sodium centate, and still some work is going on, and that's why we didn't publish these results. So with the same ligand, if we change the conditions, so we are getting a propeller shaped compound, iron 3 compound. And if you look at the K2 versus T, it is a ferromagnetic substance. And if you give the bulkiness of the ligand structure, giving more bulk to the aldehyde part and amine part, so it's turned to an Fe10 cluster. So this is the way in which it is forming. It is starting the nucleation, and it is propagating, and with the oxo bridging, and it is going on, and it is terminating, just like so. A big cluster is forming. So these are the different strategies I used to prepare the clusters. And if you check the core of the Fe10 compound, it belongs to lepidocrosite configuration. So when we are doing with the coordination chemistry, we can meet with a lot of wonders. When we are not only looking at the magnetic property, mainly we are looking at the magnetic property, but in structural aspect also we are looking in different aspects. So this is the work of Fikri. Fikri did with some chromium compound, and my student, and he got a iron 3 antiferromagnetic compound. You can see the K2 versus T here. And the ligand used is here. You can see from a diamine, and this is orthovanillin. Ship-based ligand, strata-tridentate ligand. It can be of different coordination possibilities. And this is with the manganese, the same ligand also with the manganese, and that also having antiferromagnetic coupling. And if you go to then the ligand is completely changed. We move to some other ligands, the amino antipyrine and some salicylaldehyde derivative, where we got a cobalt complex, a monomeric complex, and it is having some kind of pseudodimeric structure. And actually, these compounds are shows single molecule magnetic behavior. It's a field induced single molecule magnetic behavior. So you can see the magnetization and in phase and out of phase signaling here. This is a true signature of single molecule magnets. And then the next work is with the approaches with the spin crossover compounds. Another PhD student of mine, Joseph, was. And actually, spin crossover compound are that they have two different spin states, low spin and high spin. It can be changed from low spin to high spin by applying external stimuli. And actually, this some kind of work I read in KIT with the Professor Ruben, and this was the compound I prepared there. You can see here, N3 coordination here, and the iron 2 is here. And after synthetic, you can see here this iron 2 is coordinated to 6 nitrogens here. And this is a crystal structure of those compounds. And if you look at here, you can see the KT versus T from low spin to high spin. Around room temperature, the spin crossover happens. And it is very steep also. And this kind of spin crossover compounds can be used for display devices. And regarding this, is my student did recently, and he made the same kind of coordination environment, and he complexed with the iron. But here, instead of that compound bispyrrosol pyridine, we imparted some kind of ship-based grouping here, and pyridyl group is already there, and we gave an N6 coordination here. And this is the crystal structure of, he got it. But unfortunately, the compound was not switching. It stayed in the low spin condition. And he varied the ligand field little bit. And in the same case also, he got the somewhat the same ligand structure, somewhat the same architecture he got, but the he also got this here also a low spin condition is possible. Then another compound he prepared is submitted in the polyhedrons, and he made the ligand field variation by putting a methyl group here. If you could see the synthetic condition. So here the ligand field is varying. We are trying our best to changing the ligand field and making to the spin crossover possibility. But unfortunately, this compound also shows a low spin behavior condition. Then next work is with the lanthanide-based molecular magnetic materials. We are using the electron-rich f orbitals of lanthanides. One of my recently graduated PhD student, Dr. Mamo, was working on that. And we got some interesting results on that. Actually, I had some previous experience with this kind of things, a dysprosium single molecule magnets. I published when I was working with the Professor Pavel's group. Professor Pavel's group. And this is a dysprosium triangle prepared there. And it was published in Angwande Chemistry International, and it is highly cited in the literature. It got around 474 citations up to this time, and this is very important as far as it shows a single molecule magnetic behavior at thermally populated excited state. Actually, we try to prepare a triangular substances like this by mixing two triangles, and we try to enhance the single molecule magnetic behavior. Actually, the single molecule magnetic behavior was also increasing. This also come in Angwande Chemistry International. And regarding to Mamo's work, when we are doing by here, and here we prepared some base lanthanide complexes, and we prepared a dysprosium dimeric complex. You can see from the structure here, there are two dysprosiums are here, and this is also a ship-based complex, and it is came in New Journal of Chemistry. I didn't update the reference citation, and this is the packing of that compound. It shows also a field-induced single molecule magnetic behavior. You can see the out of phase, in phase signaling, etc. here. And we also prepared an erbium triangle with the here there's the ship. We tried to condense it with the ship base, but the situation of the coordination never allows to make ship base condensation, and it's only the orthogonal is coordinating, and a triangular formation is happening, just like dysprosium I discussed before. This is a packing, and here also a field-induced SMM behavior is observed. This is the detailed AC and AC magnetic susceptibility measurements. And again, we got a dysprosium 4 single molecule magnets. It has a butterfly structure configuration, and it also showing single molecule magnetic behavior as evident from the out of phase and in phase signaling. And this is my research group, and among that, 3 of them are graduated and 1 is still working. And these are the collaborating institute and scientists, and we have Professor Ruben from Karlsruhe Institute of Technology, Professor Boka from Slovakia. And from Slovakia, we have Professor Juraj, Professor Clarac from France, and Professor Peter Horbarik from Slovakia, Professor Linet from Austria, Professor Tepil from Badar University, and Professor Nandakumar Kalarikal from MGU, and Professor Tang from China. So thanks for your attention. Thank you.
Thank you very much, Dr. Madhu, for this beautiful presentation. So now, questions? Questions from the audience? So, Dr. Madhu, I have one question. What are the major applications of these materials?
Actually, now we are struggling with the blocking temperature, the temperature at which shows single molecule magnetic behavior.
Okay.
So the SMM behavior, single molecule magnetic behavior, that we observe at very low temperature, around 10 Kelvin, something like that, 10 to 15 Kelvins. Actually, in this particular field, the temperature, the blocking temperature is increased to 60 Kelvin also.
Okay.
Actually, if you are, frankly speaking, if you are asking, I'm giving the answer for that. If you could make a single molecule magnet at a room temperature, it will be a change for the technology. Even a single molecule can be replaced by a big bar magnet in electronic devices.
Okay. Okay, thank you very much for the answer. So if there are no further questions, then let's thank the speaker for this wonderful presentation. Thank you very much, Dr. Madhu. Thanks a lot.
Thank you, Ramesh. Thank you. Thank you.
Next, we have a talk by Professor Aragal Abebe, Bahirdar University, Ethiopia, on the conversion of toxic molecules to medicinal applications. I request Professor Reggie Philip to introduce the speaker.
Good afternoon, Professor Abebe. Hello.
You hear me, sir?
Yes, yes, we can hear you.
Yeah. Have you seen my slides?
Yes, we can see that. Could you please
Okay.
press F5 so that it goes to full screen?
Yes.
F5 on your keyboard.
F5.
While you do that, I'll introduce you to the audience.
Is it correct now?
Uh,
Are you seeing full screen now?
no, it is not full screen yet, but it is visible. We can see that very well. You can try. So Professor Abibe is, he received his PhD in inorganic chemistry from Addis Ababa University. He began working as a teacher and researcher at Bahir Dar University, Ethiopia, after getting his PhD. At the moment, he teaches inorganic chemistry at Bahir Dar University, Ethiopia. His research focuses on the modification of toxic and inert molecules for medicinal applications, the synthesis of mixed ligand metal complexes with phytochemicals as component ligands to improve their biological activities, and for the use of surface modifications for various applications, including as sensors, as well as the synthesis of new ionic liquids for electrochemistry and as modulators of material properties. Yes, we can see you now, and we can see the screen, but
Okay.
but we need, yeah, this
Couldn't
is
be full screen, right? I have
you can
pressed F.
you can yes
Now
you can try. Yeah, now I think it's okay. I mean,
okay.
yeah.
So thank you for inviting me on to present my works. I address my regards and thanks to the organizers of this international conference. I am presenting from East Africa, Ethiopia, and I have a very great love for India and Indians because it's a very long history, but I make my reasons very short and present for the audience. I have the opportunity to visit India for academic reasons and found out India to be a land of ancient literature and philosophy, where I've learned a lot about their contribution in introducing humanity with its real nature, because their philosophy introduces or teaches that the physical universe actually is just mental, not absolutely real in itself. By that, I have really, since then, my worldview is totally changed. And I believe that the rest of the world needs to consider the literature and philosophy of India. The other thing, the other reason why I love, of course, love doesn't require any reason, but one reason I love India is in the last 2 years, my country has encountered some internal instability. During that time, the Western nations were disturbing the country by imposing their interest in our internal affairs. And India stood at my country's side for the struggle against their intervention in our internal affairs. The other is my life, in most cases, is linked to Indians. My teachers since high school, physics teacher, his name was Joseph. I'm sorry for not presenting his picture, because at the time when I was in high school, taking pictures was very difficult. When I joined the university for my BSc, I got a very lovely instructor, Professor B.S. Chandravanshi in analytical chemistry, who taught me BSc and MSc. and Professor Vijay Raju, my instructor and project advisor in my SSC programs. Now, Dr. Madhu Thomas, the person who presented his work a bit earlier, is also my mentor, my collaborator, my teacher, and my friend. He is now teaching at Addis Ababa Science and Technology University, Addis Ababa, Ethiopia. The other person, an Indian person, who is very lovely person, he's very cooperative. His life is full of adventures, and he has become my model very recently. Professor Nanda Kumar Kalarakil, collaborator from Mahatma Gandhi University, Katiyan, Kerala. After I have said this much, my research interest focuses on conversion of toxic materials into medicinal applications, especially molecules, by coordinating with transition metals. Synthesis of transition metal complexes for sensor applications by modifying the surface of electrodes and sensing some toxic materials and so from real samples. And synthesis of transition metal complexes for dye-synthesized solar cell application and so on. As I have said, my basic research is focused on coordination chemistry, producing coordination compounds. Coordination compounds are, briefly, molecules that possess a metal center, usually transition metals, usually having deficient d electrons, that is bound to ligands having atoms which have a pair of electrons. For instance, such a very simple reaction tells us the production or the synthesis of coordination compounds. When we treat nickel chloride with water, for instance, water by using the electrons, the lone pair of electrons on oxygen, satisfies the electron deficiency of nickel by what you call coordinate covalent bonding. And until some rules or some thermodynamic requirements are fulfilled, the number of ligands are coordinated. In this particular case, 6 water molecules are coordinated. But this such kinds of coordination compounds are not very stable, and because of this, they can be used for raw materials or as starting materials for the preparation of coordination compounds by identifying appropriate ligands so that the desired property is obtained to meet intended applications. Therefore, coordination chemistry is about tuning the properties of metal ions using different ligands in a way that we are tuning the frequency of communication device like radio until we get the very audible and attractive voice by bringing metal ions having different oxidation states, a different elemental type with different ligands. We can produce compounds with the desired property. Now, during the coordination, not only the property of the metals is modified or tuned, but the ligands or the molecules are also modified. There are different types of molecules. Some of them may be toxic in behavior, insolubility, and so on and so forth, both the metal and the ligands. When they come together, properties like toxicity, oxidation state, stability, solvophilicity, electrolytic and nucleophilic properties are modified or are adjusted or tuned to meet the desired properties. This means synthesis of different coordination compounds with desired properties by ligand tailoring for intended application is possible. Now, one of the purposes of, or one of our work is focusing on preparing transition metal complexes for medicinal applications. While transition metal complexes for medicinal purposes are prepared, some points are taken into consideration. One point is the shape of the molecule or the medicine or the complex that is prepared. Usually, it is preferred to have flat structure like this, so that they can intercalate either in the main group or in the major or minor group, or interact in some form with especially the genetic materials, the DNA, because disease causing bacteria, for example, are causing the problem by multiplication. So to hamper or to prevent their multiplication, introducing molecules like this into the structure of the DNA prevents the multiplication or stops the action of the action of the disease-causing bacteria. When we come cells like cancer cells, their proliferation or multiplication still can be stopped by introducing flat molecules or coordination compounds like this. Now, in the previous case, in this case, you see the coordination compound formed is using platinum. These anti-cancer agents are, even if they cure to some extent, they are causing problems, side effects in the patients. This is probably, we think and we believe and we anticipate that, as you may see the periodic table here, organisms in the globe now, in the state of human beings and other organisms now, are composed of elements, basically elements in the second period. In the second period. So when elements or compounds containing elements heavier than these elements are introduced in the body of organisms, some incompatibility is believed to arise. Therefore, we focus on preparing medicines having components possibly near in the periodic table to carbon, nitrogen, and so on, or in the second period of the periodic table. Because this brings the energy compatibility issue. As you see here, platinum is very, very far away from the second period. Therefore, it is causing the problem. So, as far as possible, medicines must be prepared, or better to be prepared, by elements containing structures near to the elements in the second period of the periodic table. For this reason, we advise, or we carry out our research on preparing alternative medicines, both antibacterial and anti-cancer agents, from small molecules like this. But we have encountered that all these molecules, small molecules, as you see, all the atoms, all the components are in the second period. So biologically, energetically, it is very compatible. However, because of their structure, they are found toxic. Why are they toxic? Where they are introduced into the body of the organism because they have a very weak sigma base and strong parasitic character. They take off metals which are very essential in the cell, in the body of the organism. For instance, if we take this is a hemoglobin structure, when this molecule, even if it has a desired structure, when it is introduced into the body of the organism, it collects or it takes off the metals from the molecule and destroys the structure as well as the function in such a way, and forms such a kind of structure by destabilizing or destroying this structure and making lose its function. Now, to avoid this problem and to get the function of these molecules in medicine, what is required is to engage the structure or the part of the molecule that creates the problem by taking the metal ions from the system. This can be done by several methods. One method is engaging these coordinating atoms by metal ions in the form of coordination chemistry like this one. For instance, this is one of our works. This is cobalt chloride, hexahydrate treated with phenanthroline and formed such a structure. And when this modified structure, that is, these nitrogen atoms which are problematic in taking metal ions from the biological system, now are engaged or made busy so that they do not take off the metal of the biological system. And this complex is used to transfer this important molecule to the desired site and interact and act according to our interest. Similarly, this is 2,2-bipyridine, which is a very toxic molecule. So still, these two nitrogen atoms are strongly forming coordination compounds with the metals in the biosystem. To avoid that problem, we first engage it by alkylating or quaternization and leaving the other to be coordinated to the metal system like this. And this system transfers these important molecules to the target to perform the desired action. Now, the biological activities are tested. The free is very aggressive, of course. When coordinated like this, still one nitrogen atom is available. It is showing high biological activity. But when coordinated to the metal center, still the activity is increasing without removing any metal from the biological system. And it has demonstrated medicinal activity comparable to the commercially available gentamicin, a very powerful antimicrobial agent. So by engaging the coordinating nitrogen atoms, it is possible to remove their toxicity. Some other compounds are also designed, as I said earlier, by combining metals with different oxidation state, with different ligands of different composition, like this one. Here, for example, only 1,10-phenanthroline and water. When it is mixed with other ligands, still the biological activity is tuned in a manner we want, to develop or to generate the property we like. And the biological activities are tested in vitro, and promising results which may replace currently available commercial antibiotics, which are facing antibacterial drug resistance are replaced by these compounds. Such compounds are exhibiting numerous biological activities such as antiviral, anti-inflammatory, anti-tumor, anti-candida, and antibacterial and antimicrobial activity upon binding to the DNA. However, still there is a very big concern about these molecules. When such flat molecules are aggregated around the metal center, a three-dimensional structure is formed. This structure prevents the very strong interaction of these molecules with the DNA strand. This is believed to diminish the activity of the complex. So what can be done to make this disadvantage of coordination compounds in a better position? This is done by engaging the coordinating nitrogen atoms with a structure that may avoid the formation of rigid three-dimensional structure, so that the interaction of the molecule, the biologically active molecule, with the DNA is minimized. This is done by quaternizing one of the nitrogen atoms, so that its coordination ability is very significantly diminished. Therefore, the removing tendency of important metal ions from the biological system is diminished very strongly, and the flat structure is retained. This is done, and biological activity tests were made, and very promising results on the biological activity was obtained. When you compare with still with gentamicin, this commercially available drug, and compared with coordination compounds, these compounds showed a very promising biological activity. For instance, if we take the bromide version of this biological molecule, it inhibited 19.75 millimeter or killed bacteria, or stopped the multiplication of the bacteria. And when the ligand is changed from bromine to another, still better activity is obtained. But when we compare it with the complex, with rigid three-dimensional structure form of this compound, far better activity is recorded. This 17.25, here it inhibited 19.75, and so on and so forth. So this activity in some forms of, especially on gram-negative bacteria, which are usually creating problems by their bacteria drug resistance, can be solved by producing materials or drugs or medicines from flat molecules like this, using this structure. After saying this much about our works or our direction, actually have been indicating the direction of our work. To work this, we have collaborations from institutions and scientists. The Swedish International Development Cooperation Agency, ISP, is one. Dr. Madhu Thomas, the person who presented earlier than myself, at Addis Ababa Science and Technology University, Addis Ababa, Ethiopia. Professor Nanda Kumar Kalarkar, Mahatma Gandhi University, India. Professor Wolfgang Lienert, Institute of Applied Physics, Vienna, University of Technology, Austria, are my collaborators. And thank you very much for your attention.
Thank you very much, Professor Abebe, for this beautiful presentation, and in particular for your special love for India and Indians. So we hope you certainly get another chance to visit India, and in particular Kerala. Again,
Certainly. Thank you very much.
thank you very much. So now the session is open for questions. Any questions? So, Professor Abebe, I wanted to ask you one question. So,
Yes.
when you modify your molecules for removing the toxicity, how do you really test it out? I mean, once you have made the modifications, how do you know that the toxicity has been reduced?
Yeah, one way we checked is we let them interact with other coordination compounds
Mhm.
having the essential metals in them and mix them, treat them, and characterize them. And we found out that before and after a modification, before modification, these molecules are found removing the metal ion from
Mhm.
the other complexes.
Okay.
But after modification, we found out that the previous complexes retained their metal ion.
I see.
So in such a way, we tested we made a preliminary test actually.
Any clinical tests have been conducted so far?
Not yet.
Okay.
We are thinking to do it very soon. One is with Mahatma Gandhi University. We'll do it very soon.
Very nice.
Yeah.
So, if there are no further questions, then let us thank the speaker for this beautiful presentation.
Thank you.
Thank you once again, Professor Abebe.
Thank you very much. Thank you very much for your attention.
Thank you, sir.
Oral presentation and short invited talks are proceeding in the Narmada building. Oral presentation chess number O15 has presented and short invited talk S8 has presented.
We will resume the next 3 parallel sessions after a short tea break. Kindly note that parallel session 1, short invited talk SI 13 to SI 15, and parallel session 2, oral presentations O19 to O26 will be taking place in Narmada building. Parallel session 3 will be taking place in Sabarmati building, which is the same venue we are in at the moment. I repeat, the parallel session 1, short invited talk SI 13 to SI 15, and parallel session 2, oral presentations O19 to O26 will be taking place in Narmada building. And parallel session 3, which will be a technical session, that will be taking place in the Sabarmati building.
Another announcement. Participants staying at dormitory at Padanakad are required to be ready by and board the bus before 7:45 a.m. tomorrow. The shuttle will start at sharp 7:45. The participants are urged to strictly adhere the timeline for the smooth functioning of the conference and maintain the routine university shuttle transport unaffected. Please note, participants staying at dormitory at Padanakad are required to be ready and board the bus before 7:45 a.m. tomorrow. The shuttle will start at sharp 7:45 a.m. The participants are urged to strictly adhere the timeline for the smooth functioning of the conference.
We will resume the parallel session 3, technical session, after a short tea break at sharp 4:30 p.m.
Hello, 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 hello. Hello, 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 check, hello, 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 check, hello, 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 check, hello, hello, check, hello, 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 check, hello, hello, hello. hello, 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 check, hello, hello. Hello, hello, check, hello, 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 check, hello, 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 hello.
All participants are requested to be seated. The technical session by Atos Instrumentation will begin shortly. All participants are requested to be seated. The technical session by Atos Instruments will begin shortly.
Please note that parallel session 1 short invited talk SI13 to SI15 and parallel session 2 oral presentations O19 to O26 is currently taking place in Narmada building. Oral presentation O18 has ended. Participants O19, O19 to O26, O26 has to be reported in Narmada building.
Hello? Hello? The technical session by Atos Instrumentals will begin shortly. Participants are requested to be seated. The technical session by Atos Instrumentation will begin shortly. The participants are requested to be seated.
Participants of oral presentation O19 to O26 kindly report in Narmada building.
To begin the first technical session, we have Dr. Trinath Banik from Atos Instrumentation.
Yeah, hi, madam. Yeah,
Hi, sir. You can start with your presentation.
yeah. Okay. So I'll just share my screen. I think my screen is visible, right, ma'am?
Yes, sir. It's visible.
Yeah. Thanks for allowing us to present our product today. I just wanted to thank the conference coordinator and all the distinguished scientists, researchers, and everyone present in the conference. So myself is Trinath Banik from Atos Instruments Marketing Services. So I just present our product today in this conference and have a brief of whatever the product we have. So starting with our company portfolio or company profile, We have around 860 installations in India, over 200 institutional coverage with our different spectroscopy products, lasers, cryostats, and other equipments. And over 83 cities, we have the presence of Atos Influence Marketing Services. So just to start off the company portfolio, I just wanted to say that we have started our company back in 2013. But our CEO has the experience of around 25 years of in the scientific market. So with the spectroscopy, with the CCD detectors, with the